So good afternoon, everyone, and welcome to today's webinar in the HELIOS HemaFAIR webinar series. We are excited to host Annika Ahlford, Director Product Manager of DVYSR, a company specializing in advanced molecular diagnostic solutions, who will present a talk titled Development of Molecular Tools for Diagnostic Applications within Academia and Industry. This webinar is part of our effort to strengthen engagement between academia and industry and to highlight advances in the molecular diagnostic field of hemoglobinopathies. Please note that this webinar is being recorded, and we are very thankful to have Annika joining us today and sharing her expertise and experience between the two sectors. Thank you all for being here, and I hope you enjoy the webinar. And with this, I give the floor to Annika.
Thank you so much. Yes, I'm very happy to have the opportunity to introduce describe my journey and my experiences with the development of molecular tools. So my presentation will include some introduction to the Swedish life science legacy. Also, it will touch on my academic journey. I will introduce DVYSR and our value proposition. I will describe our single tube NGS concept and also describe one application. And finally, mention some innovation within DVYSR. But let's start with the beginning. So I did not have really a clear career plan when I was growing up. But then I met the inspiring high school teacher, which many of us do. He was a former scientist from the Karolinska Institute, and he introduced me to molecular biology in 1997. So I got very interested, and I was looking for courses on the topic, and I found a master's program in biotechnology in Uppsala at the Swedish Agricultural University. And I started my undergrad studies, and really the learnings from this period was really to learn the theory, the hardcore genetics, plant genetics, agriculture model at the most, but also the molecular biology theory and also the practice. And I was also introduced to the infrastructure of the region, which really was very important for the next step. And I would like to describe this a little bit, the so-called Uppsala-Stockholm corridor. It's a little bit of a biotech valley, and it's actually one of the most dense life science clusters in the world. And the scene was seeded by some large pharma companies, Pharmacia and Astra, who today are split, and they are so Astra is together with Seneca, still today available, and Pharmacia was split. It was Pfizer, and it was Thermo Fisher, and then also Cytiva. So this then also, this was the legacy, and there's been many startups in the region. And this was partly due to a very high functioning ecosystem. So researchers can easily transition discoveries into commercial entities via dedicated incubators. So we have 3 major universities in the region. It's the Karolinska Institute, Uppsala in Stockholm. We have Uppsala University and the KTH Royal Institute of Technology. And something which is very important to mention is that this was fueled the success in this region by the teachers' exemption. And you can wonder, what is that? But it means that the scientists in Sweden, they own their own intellectual property. It's not the property of the university, but the IP is owned by the researchers themselves. And this really provides incentive for academics to launch startups. So all of that, I would say, creates a very creative environment. And Uppsala is often referred to as the city of methods. And focusing on research tools and diagnostics. So very strong in diagnostics. And I actually became then very interested in methods. Most scientists are more interested in the endpoint. They would like to use tools to solve their endpoint problems. But I got very interested in the tools themselves. And to collaborate in various different use cases with these different molecular methods. That was what I got very inspired by. And so it means, of course, that you need to collaborate with very different type of people. So I was also inspired by this ecosystem and all this intra and inter institutional collaborations. So I stayed in the region and I started continued my academic journey then at Uppsala University. So my PhD studies were conducted in the group for molecular medicine. And it was actually a group that was part of the academic hospital in Uppsala and led by Professor Ann-Christine Syvänen. So this was quite unique that we were sitting really next to the clinicians and doing science together. So, and that was, I would say, was one of the recipes for success. And the learnings from this period was also really to continue the practical and handicraft of molecular biology. So at the time, we were developing our in-house based systems. So my professor, Ann-Christine Syvänen, pioneered actually the single-base primary extension principle and used for genotyping. And genotyping was hot at this time. So we had our own microarray-based technology that was at the time pretty advanced. We could do, we printed our own microarrays and rubber grids to separate samples. So it was multiplex. We could analyze a couple of hundred SNPs for up to 96 samples within a single test. So it was very cost efficient and quite high throughput. So I learned a lot about the molecular principles and the tools available at the time. And we were part of developing this. And as you can see there, this was took up a big part of my thesis. But I was also really fortunate to be that part of our research group was also this core facility. So we had access to commercial systems for SNP genotyping from Illumina. So something we did at the time was to perform together with the clinicians association studies for different diseases. So very handy to work closely with the clinicians. And we were very fortunate to, in 2007, have the first Solexa genome analyzer, which was the precursor to Illumina sequencing. So I think we had, if I can remember correctly, it was facilitated by a grant of €1 million. And this was really exciting times. So that was also part of my learnings and the fantastic opportunity to get to know these tools and understand, even with the commercial versions. I was then also fortunate to have my second supervisor was Professor Mats Nilsson, and I started my postdoc studies on the other side of the street at the Department of Immunology and Pathology in the group of the famous Professor Ulf Landegren. And they had together pioneered many different principles that were actually not PCR-based and not the standard. So I learned about other molecular principles and techniques, so-called padlock probes, rolling circle amplification, proximity extension and ligation, etc. And these tools were, so you can see a picture there where a padlock is locked over a mutation and sealed with a ligation, and only if it's circularized, it can then be amplified and generate a long concatenated molecule that can amplify the signal needed for readout. It was very, very sensitive and specific way of amplifying very even low abundant signals, and a very sensitive way of measuring DNA, but also proteins, both in solution, but also in situ, or like in cells, but also in tissue. So this was quite exciting. And I would like also to mention that the learnings from this time was, in addition, also so I could really be exposed to the entrepreneurial vibe here in this group, because it's not so common, but there are over the years more than 10 companies that have spun out from this research, and you have probably heard about Olink Proteomics that was acquired by Thermo Fisher not that long ago, and also Halogenomics was acquired by Agilent. So there was a lot of great successes where these early tools were implemented and commercialized and scaled up. So it was really, really exciting to be part of this. I have to mention also, during my PhD studies already, I got interested in going abroad, and I applied for travel grants for a research exchange in Boston. And I was really fortunate to be able to join the Church Lab, the so-called Church Lab at Harvard Medical School. So if you are talking about molecular tools development, you must mention George Church, because he's really often referred to as the founding father of the genomics industry and also synthetic biology. And he's really a guru when it comes to molecular methods development. And, you know, we could talk 1 hour about George Church, but I just wanted to list some of the quite spectacular things he has contributed with. He was early on very important in developing sequencing technology and also molecular barcoding, and that allowed indexing of samples and the molecules so that you can multiplex and analyze things in parallel. And one of the bits I wanted to show this, one of the bit spectacular things is his attempts to work on recreating the woolly mammoth. So he's really involved in a great, very broad initiatives, everything from storing DNA data to different types of synthetic biology approaches. But why I'm mentioning him here is that around the millennium and the first years there after the millennium, they were also focusing on next generation sequencing. And they had developed a platform that was actually based on the principles of the Uppsala scientists, Mats Nilsson and Ulf Landegren. So I was there to learn and to further develop, and it was we applied and also further developed these processes and implemented for different applications, for example, RNA editing, which was quite interesting. And the learnings and what I really think is important from this time was also to see this kind of network and build my own network, because the Church Lab is famous for its alumni framework. So it's really a very inspiring and nice network to be part of. And so this is something I would like to stress and maybe recommend the students in the audience to do not hesitate to apply for travel grants. They're decently easy to get. And these kind of research exchanges can be really rewarding. So, so my last part of my academic career was spent at something called SciLife Lab. So I followed Mats Nilsson during my postdoc to Stockholm, where we were part of Science for Life Laboratory. And what's that? So that's a hub for molecular biosciences that was founded in Sweden. And it's a national research infrastructure. So it really includes almost all large universities in Sweden. And it offers then service-based infrastructure that everyone can take part of. So genomics platforms, proteomics, spatial biology, and also platforms within drug discovery. So it's a broad network and infrastructure focusing a lot on bioinformatics, etc. Everything that research. So the point is to facilitate this type of tools and avoid that everybody must have the so it's because everybody does not have the opportunity to establish that in their own lab. So it's a way to facilitate these kind of tools. And it's a nationwide structure, but there are 2 hubs in Stockholm and Uppsala seen in the pictures, and we are part of the Stockholm hub. And not only was this very important for scientists, but also it's, again, an incubator for startups and an accelerator, because also the startups can utilize these services, and it's really great. And this also has fueled many startups in the field and in the area. So what did I do here? Well, already actually throughout my PhD studies, postdoc, and also my last time here, I was actually involved in many national and international projects applying molecular biology. And as I said, working with tools, you work with very different people. So many clinicians, but also engineers. We worked with lab-on-a-chip systems, very exciting, working with microstructures for point-of-care testing. So really broadly, everything from human and Drosophila model organism in my thesis to doing molecular analysis in a cell phone. So it was very broad. So I have now described a little bit that I was fortunate to work with the right people when it comes to method development, but I also was at the right time. And I need to emphasize that a little bit more. I already mentioned that we had access to novel type of technology, many array-based at the time. It was arrays for expression analysis, but also for SNP genotyping. But absolutely what kind of shifted, the next big shift in technology development was the revolution of the next generation sequencing, NGS systems, and the introduction of the commercial systems around 2005. So in the old days, we were used to working with the ABI systems, Sanger sequencing, working horses, still gold standard, to enable sequencing the human genome and great efforts. And then suddenly you have all these instruments coming. And they pioneered, so it was the genome analyzer by Solexa, which is now part of Illumina, and the Roche 454 that came there in around 2005. And you can really see how here, this you've probably seen, but how the cost per genome, per human genome dropped radically after that. And this is now how it kind of first followed Moore's law, and Moore's law is the standard in rapid technology development, and then it dropped dramatically below even Moore's law. So, and how was that possible? So it was because we got the massively parallel aspect. So before it was more sequential, and now millions of molecules can be analyzed in parallel on surfaces. So it was also miniaturized. So using actually microfluidics and integrated systems. And then also there was a digital integration. So there were cameras and digital processing of the data, which facilitated this, and which kind of, it transferred from a biological process to a more digital process. And this really together all led to this revolution. And I just wanted to show again, so everything kind of ties together. Here's the picture of this first Solexa instrument that we had. This was the one we had in our lab in Uppsala, where my professor actually was also part in the principle. Everything goes together, and it was a super bulky machine. You had to put oil on the surface yourself and put the cover lid, and it was really very exciting times, but it was quite hands-on still. And this, I would say, there's many sequencing technologies that have come out, and now are again very exciting times. At the end there of the list, you can see the Oxford nanopore Me9 that was launched about 10 years later, and there's a lot of different principles coming out now and further developing and towards third generation sequencing. But I just need to still emphasize that the Solexa system pioneered in the UK was really what really made it possible to reach. And it's still, Illumina today has a very dominating role here in the market. So with that, I want to finalize just to really understand how big this is, that when a technology outspaces Moore's law, it creates a groundbreaking shift in what is possible. So today, actually, we have come down to a $100 genome, and it takes hours to days to perform a sequencing of a whole genome. And it's available to individual clinicians and not only to large consortia. And we work towards precision medicine. So it's really amazing. And that concludes a bit the first part, which is my academic path, and kind of ties together my experiences with the timing. And now I will move to the DVYSR story. So, right. So about 10 years after the first sequencers, the commercial ones, hit the market, I then and then those first 10 years, really, the sequencing technology was mostly applied within the context of research. But then in 2015, I got the opportunity to move to industry and start working with DVYSR, and to start actually implementing this technology in the context of clinical sequencing. So first, I would like to present DVYSR a little bit. So we are a Swedish company, but we're operating globally, and we are we have 20-year experience in providing solutions for genetic diagnostic and clinical research. And I would like to show first a bit the value of correct and fast diagnosis to really get you to understand what is important when you come to industry. So the patient is important here from our perspective. So waiting for the test result is really stressful experience, and the outcome can be life-changing. So DVYSR, our mission is to be a pioneering leader of diagnostic solutions and to provide fast, accurate, and easy-to-use solutions to labs worldwide. And our vision is a world where personalized medicine is universally available thanks to simplified and reliable genetic tests. And this is really the key words that I will come back to. So as I said, Swedish company, around for over 20 years, we have our headquarters in Stockholm, where we have product development and production facilities. We have subsidiaries throughout Europe and in the US. We are specialized in easy-to-use kits for complex DNA testing and also software. And we focus on routine diagnostic applications, and we use different type of technology platforms, are compatible with different technology platforms, with a focus on NGS technology. And we have, since 2006, launched over 25 CIVD labeled kits, and we are fully compliant with international standards for medical device manufacturing, and we are shipping our tests worldwide. Some more numbers. Just want to show you a little bit what DVYSR has the last 20 years. So it was founded in 2004. And we have, from the start, worked with certified products for the clinical and diagnostic market. Into 2021, we were listed in the stock market, and we have a big focus on developing in North America. And we have, in 2023, we opened a CLIA certified laboratory for providing services in the US market. And as we have been growing, we also have now partnerships with some of the major biotech companies, such as Thermo Fisher and also Illumina. And we have products within different clinical areas and diagnostic areas. So the legacy and the majority of tests are for testing of hereditary diseases to provide early treatment and better treatment of rare diseases. So we also have a smaller group of oncology products, mainly also for testing for hereditary cancer. So it's, but for oncology, for breast and ovarian cancer, mainly focusing. And we are also actually supporting not only the hereditary cancer, but also supporting in the treatment stratification within oncology. Then we are actually also working with post-transplantation products, and I will come back to that a little bit later. Okay, so as I said, you come from being a scientist and you come to DVYSR, and you're used to working always with the most novel things in the market, in the field, because that's how you have publications. And then you come to the reality where you want to implement and really make a difference and really ensure that it can be applicable the technology in every lab. So what is that about suddenly? Well, it's to have simple, fast, and accurate testing solutions. They have to be robust. They have to fit with the cost, requirements, etc. So DVYSR has, from the start, worked on eliminating tedious protocols, creating end-to-end workflows and solutions, and streamlining workflows, and to really create the products that would improve, on one hand, the lab efficiency, so the lab part, but also simplify data analysis. And to provide cost-effective and result, because healthcare is it's dictated by resources, what we can do, and resources are aligned with guidelines and reimbursement, etc. So it's important to have cost-efficient and effective solutions that actually provide actionable results. So we want to provide novel and innovative products which still provide actionable results that are clinically relevant. And of course, we want to benefit the patient. So we have focused on developing tests that are easy to use, fast, and reliable. And this is our tagline, a new level of certainty. So it gives the certainty and the reliability with also an innovative angle to it. And I think this is maybe the place where I can share my journey at DVYSR. So I started as a project manager and technical lead in the R&D department. So I was part of launching our NGS products, the first in 2016. So I productified the prototypes. It was a startup phase. We were around 20 people at the time. And I have to say, one thing that was also a little bit different to academia was that there was a clear team effort. That in academia, you're a little bit more working in parallel. And here in this company and startup phase, especially, it was really a big team effort to launch these NGS products. And I have stayed at DVYSR but continued to are now part of the commercial operations. Started as a product manager in 2021, and today I'm leading the product management team. And for me, this is really perfect. It's working with the commercial and the product, the commercial side and the products, and together with the customers and the labs, even the patients. But you have your leg also in the technical world. So for me, this is really perfect. And so I kind of mentioned the value proposition of DVYSR high level, and this is how we now implemented this for the NGS approach. As I said, waiting for test results is stressful, and also producing and analyzing genetic results have traditionally been complex and laborious and time-consuming. And not only that, but also a risk of errors. That's why it's so important to simplify, standardize, and provide validated protocols that work every time. So we redesigned what used to be multiple steps, multiple tubes to a single workflow. And we introduced something that we called single tube clinical NGS. And this is then important for time and cost savings of the lab, but of course, most importantly, giving each patient a correct diagnosis in the shortest possible time. And now I would like to go through a little bit what that means in detail. So we deliver simple and fast tests. As I already mentioned, we have fully validated the end-to-end workflows, which means we support the customer from the start to the end. And this is based on targeted amplicon sequencing. So we have both developed target library generation kits that are then sequenced, and then we have supportive software to analyze the data. Zooming in a little bit on the library preparation part, this is where we where our, let's say, legacy competence lies, we have then developed this workflow with the key features are listed here below. So it's 1 tube per sample. So it means that we first have a multiplex amplification in a single tube per patient. And it means that very little material is needed and there is no risk of mix up, etc., when you have to split samples. So this is critical. Then we have pre-dispensed indices and material for barcoding the samples. So once you generated your library and amplified all the regions of interest, you go to barcode each sample. And this is then these features that we have, these pre-dispensed and dried indices in a plate format, really simplifies the testing. And then it's possible to pool all these samples in a single tube before bead purification, which is really a big advantage and which is still a very strong competitive feature. And all in all, this leads to a very short turnaround time, total time, and very little hands-on time. Right. And what did then facilitate this multiplexing in a single tube? Well, as maybe you know that multiplex PCR is not so easy. And it's often hindered by, if you add many fragments, they start to interact with each other, the primers, and you will get unwanted products. But we have a proprietary design which enables multiplexing in a single tube. And this is here illustrated by exon 11, where we have 39 overlapping amplicons covering the exon. And you can see here the number of exons that are included in, the number of amplicons in each kit here listed. So fine, we have solutions to enable simple tests, but also, or approaches, but also we need reliable results. So we have this overlapping design, as I said, and that enables like a very high quality. So it gives you a double coverage of the regions. So it enables the detection of primary site mutations, and it gives a more robust detection of variants. And you can identify if there is a dropout. We have paid a lot of attention to both in the design, but also QC and development and manufacturing, to uniformity of coverage. This means that you have a robust variant detection, but it also means that you can minimize your costs by maximizing flow cell capacity. It means you do not have to over-sequence some parts to be able to amplify well enough the low-performing ones. It's very even. And a very important feature is that we have focused on detecting copy number variants. So these are larger structural variations that are more challenging to detect than simple point mutations. And we use 2 approaches. So both something called coverage-based analysis, which is more like the standard NGS way of detecting. So you have a way to, between samples and within samples, compare the data amount, and you can see if there's a region where something has dropped, which indicates a deletion, for example. But we have also something more, a direct genotyping approach of deletions, which is called, or which we call direct detection. And it's facilitated by gap PCR principle. And this really is a way to specifically detect specific variants, deletions, and that adds precision to the testing. So these 2 complement each other, and that really makes a difference and provides robustness. I want to just briefly show here this direct detection. The principle is that you design primers that span a breakpoint, which can be very far, like this can be a large deletion of 5 kb here in this example. And only when there is a deletion happening, and this part of the DNA is deleted, these come into proximity and allow for an amplification of a small fragment. And only then you can detect these specific breakpoint products. So this is how it works, and we have implemented this to be in the same tube. And here you can see some of the products where we have implemented this. Right, so what is, however, the most important is to benefit the diagnostic laboratories. And I would like to first mention some major categories of DNA analysis in the clinical context, or some of the major. So as we said, diagnostic testing is the most classical one, where you confirm the diagnosis. Maybe there was another test, non-genetic test, and you confirm the diagnosis, etc. But also today important with treatment stratification. So if there is a symptomatic patient, which has it's good to confirm what mutation do they have to actually guide targeted therapies. And in the next step, actually also monitor treatment response. So that can be important in a scenario if there's a disease relapse, and also to define minimal residual diseases. So this is evolving and becoming more and more available in different contexts. Then there's you can also use genetic analysis in screening scenarios. For example, carrier screening for reproductive health. For example, asymptomatic couples that to avoid at-risk pregnancies, but also cascade testing of families of identified. If you have an index patient, then you are testing the families to find carriers, or maybe also find patients with mild symptoms, etc. Then another application or use case will be newborn screening programs that are often national or centralized, where a heel prick test is taken from the baby the first days of life. And this is also, there's a big spread or a variety what diseases are tested for. And more diseases are tested for and conditions, and more and more genetic analysis is used. So we are supporting with this type of different use cases. And I would like to just briefly, because of I'm running out of time, I will skip pretty fast through this, but I know that many of you are interested in this use case and this application. So on the 31st of March, my colleague will present our newly launched IVDR product called DVYSR Thalassemia V2. So I can recommend a webinar, a launch webinar that will be offered through our channels on the 31st of March. But I want to just briefly mention this. So thalassemia and sickle cell disease, that is one application where genetic analysis can be important for the screening, follow-up, and diagnostic purposes, for example. So it's a group of blood disorders caused by abnormal hemoglobin production, and there are different mutations leading to disease, and depending on what mutations, then the symptoms and severity of disease varies. A common symptom is anemia. So conventional testing for genetic analysis has been generally the third level test. And that is has conventionally been performed with a patchwork of different standard molecular methods. Very good each, and very usually cost efficient, but limited and targeted. So what the principle has been to work in a serial manner, start with the most prevalent variants in the region, and then continue to test. And for the bulk of the patients or the screening cases, this is relevant. But you can see here the process that this has led in many cases to quite complex clinical workflows. That testing is done for a certain mutation, then there's a new referral from the clinician for another testing, and this can continue up to a couple of months for some variants. So, and also there's a risk of unidentified variants, because usually the testing stops when the first variant is found. So also you have this sample, and if you have a testing and you go back and forth requesting a new sample and so on, there's a risk of mix-up. So, and handling multiple tubes and protocols. So even if every protocol is very standard and cost efficient, the total requirement for the lab to validate, maintain, introduce, and keep personnel active is pretty large. And so what we offered here is to add a more comprehensive test that can give a 360 view to kind of give an overview and a profile of this patient and solve the majority of cases and pinpoint where follow-up would be needed. And that's what we are now proudly presenting as DVYSR Thalassemia V2. It has been on the markets for a year now, but now we just recently launched the IVDR certified version. So what the V2 means is that we have worked on, we have included more regions in the beta, so the target region is expanded. We have worked a lot on improving the robustness of the CMV analysis, especially for these common copy number variants, the alpha 3.7. And we also have included beta modifier detection. And of course, we're still working with the streamlined workflow, which is our signature, which is very important. So even if it's more complex, we try to be as simple as possible. So this is the regions covered. I will need to continue now, but as I said, more to come soon on this from us. So you just register and listen in soon. And I need to stress that it's very, for thalassemia, there's a lot of clinically relevant and rather common copy number variants. So it's very important to support with this dual approach that is robust in detecting them. So we're proud to say that we have direct detection for 19 large deletions with characterized breakpoints. And we then are also confirming these results with the coverage-based analysis, which is using a pattern recognition algorithm. And we have added an additional 50 patterns to support for analysis of copy number variants. Very important to stress here, the software. So it's important to support with simplifying the analysis, because so this is quite complex genetics, and it's important to standardize and to really make a user-friendly way of analyzing this NGS data. So that's something we worked a lot on. So we provide QC information, variant lists, and copy number variant plots. So we have links to relevant databases, and really kind of trying to offer an integrated analysis, so to avoid the need of having bioinformatic expertise in the laboratory. And also that the targeted analysis is important to minimize the handling of non-relevant variants. Okay, and here in the plot you can see an advanced or a quite complex deletion. Compound heterozygous. So, just to summarize, we have an assay with comprehensive detection, yet targeted with a simplified workflow, a very user-friendly software. And actually, from just recently, we have a agreement for reagent rental together with Illumina, so we can also support in setting up NGS in the laboratories to also kind of bridge the hurdle of introducing the NGS part. So please tune in and listen to my colleague about our newest product release. Just after stating that we have something simple, reliable, and that it actually benefits the clinical labs, I have some some quotes from users, and I'm very proud that our value proposition holds. We have many quotes stating that it is simple and very sensitive. They get robust results, and yes, we are really happy to work with our users and further develop the products. Now, I think I am out of time. Just want to say, being in a company, I cannot disclose everything we are working on that is innovative, but DVYSR, of course, try to do the next, give certainty and innovative solutions for routine diagnostics. Sometimes we go a little bit beyond and work a bit with emerging markets. sometimes it's more working with the standard testing in the human genetics labs. So here are 2 examples now where we actually are working a bit with the progressive areas and emerging markets. So we work with cfDNA as a biomarker, and that is something we do within the post-transplantation monitoring space, both for so we have for organ transplantation monitoring. And it's a non-invasive way of detecting donor-derived cell-free DNA in the blood samples of transplant patients. And this is important because it can be an early indicator of graft injury or rejection or relapse. And it supports clinical decision-making and tailoring of therapies. And then one more example is our new product that we launched last year for genomic blood typing to complement serology testing. It's a comprehensive blood typing test for testing in patients, which is important for more accurate and complete antigen profiling. And that improves transfusion safety and reduces the risk of alloimmunization. So very important for complex patients, which and also those that are transfused regularly. And for example, sickle cell disease and thalassemia patients are one example of these patients. But also, I mean, more and more also being able to screen and profile donors, identify rare or clinically important antigen variants, and help blood centers better match donor and patients, which, you know, are things that are very important. We have still the pandemic in mind. We have, so it's important to work where serology is not sufficient or ambiguous, and in use cases where genomic can add value. This is really important. So with that, sorry it was a bit long. I would like to just say thank you. It was a quite diverse, broad presentation. If you're interested in connecting with me or with us for different reasons, you're very, very welcome to talk more about DVYSR or collaborating research exchanges. I'm very happy to, and I'm of course happy to take questions now.
Thank you, Annika. Thank you. It was very interesting to see your path from academia and how you ended up in industry and how now DVYSR, from all that development, has ended up now to working with modifiers and personalizing diagnostics. Petros has a question already. If anyone else has a question, please raise your hand. Petro?
Hi, thank you very much, Annika. It was very interesting. Especially, I enjoyed the journey that you presented, because one of the aims of this lecture series is to show to people, you know, that there are many different paths in career paths. And sometimes we have in our head, you know, a single path, and we get disappointed if we don't achieve it. So thank you very much for this. So my question is about how you update your products when there is emerging new evidence for, you know, new, what I have in mind is genetic modifiers, for instance, because when there are new modifiers that come up and are validated in research settings, how does the company integrate this on time on your product so that people can benefit from it? So what is the process, basically? Thank you.
Yes, good question. Of course, as I said, when you are in industry, you have to also see a bit of a return of investment, and it's nice to have, and what is in the guidelines. So of course we are evaluating. Of course we launch products, and there's a life cycle management. So we collect feedback and we try to understand, are there trends towards new guidelines, etc. But one cannot change frequently, but we can, of course, we must update and be also relevant. So, but it is challenging that these guidelines are quite different only within Europe, and going globally, it's not simple, and especially with more strict regulatory requirements in being both within Europe, but also we see in Asia more strict regulatory requirements in many markets. So it is a balancing act. but the way to do is to communicate and then have feedback from the market and try to understand when is the right time. That's the goal of, or the job of a product manager, to understand what products and when, and exactly what kind of translate it to product, the science to product. So yeah, so it's a bit case by case.
Thank you.
Thank you. And Jan, you have a question as well.
Yes, hello Annika.
Hello.
That was a very, very nice talk, and it was very encouraging to see your journey, because I also have a strong background in methodology. I think all people from my generation probably do. I can see Case Hutfeld's on the line as well, on the call. So, you know, we had to, I did my DPhil way back the end of the 70s, the beginning of the 80s, which means that at that time one had to make that, you know, our own buffers and understand the chemistry behind all the reactions. So it was a long, long time within my career where things became in the form of a kit, and you could do things more automatically. It's only, in fact, very recently in the hemoglobinopathies that, you know, we've got such reliable protocols like the DVYSR protocol that we can say that, yes, our in-house protocols can be replaced now by a one-step kit, because until now we didn't have this this support, I don't think, from the industry. So that's really great to see, and it was very nice to hear your journey. A couple of questions, more technical. For the minimal residual disease, what sort of sensitivity do you go down to?
So at the moment we don't offer that type of products. Yeah. So, but it's more to kind of illustrate. So we are working with the supporting in the transplantation. We are working with the relapse part. But at the moment we do not have a product for minimal respiratory disease. Yes. But that was just want to state that what type of testing is relevant. Yeah.
Okay, and one very quick question. I saw there were a huge number of amplicons on exon 11 in the BRCA gene, one gene, I think. How long is this exon?
It's one of the biggest. I mean, So that's a good question. Exactly how long? I'm not sure now.
or maybe the other way around. How long are your amplicons when you do your PCR based?
Yes. I mean, they are right. So they are around 200 on average, and of course, I would say, like you said, a standard amplicon would maybe be covered by three amplicons or something like that. So this is, of course, something quite big. Yes.
Okay. Thank you very much. Thank you very much.
Thank you. I just think it's interesting, your comment. I wonder if things get super easy if we start doing everything ourselves again. Everybody starts manufacturing their own oligos in the future. Everybody has an AI instrument doing that. Who knows? Let's see. Goes back again. Ships.
Yes. There's also technicalities of standardization and so on, which are quite exacting. So unless AI robots can do it all,
let's see.
I think you'll still be needed.
Yeah. Thank you.
Thank you, Jan. And Kees, we have a little bit more time for one more question. Kees?
Yes, thank you very much for your very nice talk. We are presently using the DVYSR kit for our hemoglobinopathies diagnostics, but I was wondering, are you also developing a kit for hemophilia? Because there are some, let's say, challenges. 30% of the hemophilia A cases are because of an inversion 22, an intron 22, and this inversion is very, very difficult to detect by, let's say, common techniques. Is DVYSR also thinking about hemophilia?
Yes, so we have been thinking about it. At the moment, it's not a clear, it's not in the roadmap or a clear timeline for any launch of something around hemophilia. But you are right that now I didn't mention that, but what we have been working on in other assays is to be able to support there's specific analysis of complex structures, also pseudogenes. So we have this PMS2 specific assay for Lynch FAP, so colon cancer testing or colon cancer syndrome testing. So we have worked with solutions for where we implement very complex structures. So that is a little bit our signature, absolutely, and kind of to package them and simplify it, the complex structures. So yes, we are discussing it, but that's probably where I can stop. I have to stop today. But let's continue to discuss it more, Keith.
Thank you.
Okay, I have one final question, more general and less technical. You mentioned that you do accept visits from students at your company. What is the best way for anyone interested in the audience, if they would like to make a visit, for them to apply? Is it like internship based, or they can reach out directly to DVYSR if they're interested?
Yes, I mean, we have had interns. So I think it's, yeah, you can always reach out to me, or I will connect you also with the right people. I think also one should not be shy in exploring like these research exchanges. I think that is something I really like. You know, if you write your letter of invitation yourself and you can get a professor to approve it, it's not so difficult to be able to come to the top labs to learn exciting things. But absolutely, we have accepted the interns before. So yes, connect. We do not have a specific well-established program, so it's a little bit based on the resources available, etc. at the moment.
Perfect. Thank you very much. Thank you everyone for attending our webinar, and a special thank you to Annika again. Thank you for this really, really interesting presentation. The webinar will be available on YouTube in due time, and I'm sure many people will attend the upcoming webinar by DVYSR as well.
Yes, I did not have a formal slide now, but you're very welcome to attend. Maybe I can share it with you if somebody's interested.
Of course, yes. Thank you, everyone. And thank you, Annika, again. Have a good afternoon, everyone.
Thank you. Bye-bye.
Bye-bye, everyone.
Bye.